मौसम खराब
So, good morning, Sharvari. I think you are the only one in the class at the moment. Um, Anyhow, we will start our class within next 5 minutes. It's already seven within 1 or 2 minutes. So we have left with the carbohydrates in our previous lectures. We didn't touch any part of monosaccharides, disaccharides, polysaccharides. So we might touch that part today. And then all the cycles also related to that, glycolysis, gluconeogenesis, and TCA cycle, ECA cycle, all these cycles will be taken care. Cool. Okay, we can start now, I guess. Um, Earlier the wake up early, start early. So basically, carbohydrates, as we have discussed so far, your proteins a lot, amino acids. We have discussed about your fats and lipids a lot. Now, third part was left actually from the earlier game. That is your carbohydrates. Yeah, their carbs are also very important to be discussed. So, which contains in food such as pasta, bread, like roti that we are eating mostly. That is your carbs. That actually provide energy to our body. That's energy that we require for our as a fuel for the whole day. So they are broadly defined as a polyhydroxyaldehydes or ketones and their derivative substance that yield of these compounds. So they're composed of carbon, hydrogen and oxygen, and the functional groups includes their hydroxyl group. And if they are having this sugar, then we add O's at the end, OSE. So, as in nature, they're abundantly present. So, in the plants, energy from the sun is converted to CO2 and water into the carbohydrates, glucose. So, in plants, their main energy source is also the glucose that they are using, which is a kind of a carbohydrates. So, many of glucose molecules are made into long chain polymers of starch that store energy, and about 60% of the foods in our diet consist of carbohydrates only. So we day to day life we are eating bread, noodles, pasta, that you momos that you having the craving in the evening. It's that's actually not wheat. That's maida, um, wheat flour. Yeah, then potatoes, rice, all these contains your carbs. So other form of carbohydrates are also called as disaccharides, which includes a sugar, normal your table sugar, and lactose in the milk. Then the milk that we're having this lactose in the form of as a disaccharide that's also present. Okay, so during digestion and cellular metabolism, these carbohydrates they are all whenever go inside our body they turn into the glucose. So that's why whenever somebody is feeling weak or they get faint, we instantly give them to drink either glucose or we put the drip of glucose so that the food that we are giving it doesn't have to digest anymore and the person could have directly the source of energy that is your glucose in the body. So whenever this glucose get inside our body, it's get oxidized and provide energy by from the carbon atoms for building molecules of proteins, lipids, and nucleic acids. And in plants, polymer of glucose is called as cellulose, which builds a structural framework and has very important uses also. So the wood in furniture, the pages in your notebook, the cotton in our clothing, they're all made up of cellulose actually. Yeah, so furniture, notebook, cotton, everything is actually at the end of your carbohydrates. So what are the functions of carbohydrates in general? So they are measures of an energy as we have discussed, structural plant of your plant cells, an immediate source of energy in the form of glucose, and could be stored in the body as a reserve energy, as a glycogen. So that they say the person could stay, uh, let's say 5 days at least being hungry without food, but not after 3 days without water, because our body is made up of 70% water, and without water, our body will dehydrate and cell starts to go into the senescent state. But in the presence of but still body have this fat they can use it, person will not die without eating but because of water it will die. So some forms of carbohydrates in our body, so glucose, so basically the glucose structures, it has 6 carbons here we can see, 1 2 3 4 5 6, and all these from carbon 2 to 6 they're all having this hydroxyl group, OH group. Uh, and then on the first carbon is your aldehyde group attached to it, COH. Then there's a galactose, which also having the same thing, 6 carbons, 2, 3. So basically, you can see the main difference is happening that the OH is here, then OH is there, and then OH is there, then OH is there. But here in the galactose, on the fourth carbon, OH is on the left hand side, In the glucose, it's on the right hand side. Then there's a fructose. Fructose is made of also 6 carbon atom, but in the second carbon it has this ketone group, this double O oxygen bond. Yeah. So that's how the main difference between your different sugars looks like. So let's see how we can classify these carbohydrates. They are classified according to their subunits, how much units they are made up of. So that is monosaccharides, oligosaccharides, and polysaccharides. So oligosaccharides could be of three types: di, tri, and tetra. Yeah, disaccharides, trisaccharides, and tetrasaccharides. So monosaccharides are simple sugars or compounds which possesses a free aldehyde group. They have the CHO group on the top or ketone group. Like in the we have seen um, this ketone group and a aldehyde group and two or more hydroxyl groups. So they are the simplest sugar and can be fertilized certain smaller. This is the one of the major smallest units, so it cannot be divided further. Yeah, so they contain a single carbon chain which is classified on the number of carbon atoms, and they possesses aldoses, ketoses depending upon their groups. So monosaccharides, they are classified according to the carbon atoms. So triose, tetroses, pentoses, and hexoses. I mean, these are kind of sugars actually. That's why the O's is at the end in the sugars, you know. So whenever there is a sugar in the sugar, at the end your O's is added. And the structure formula is as follows: C3H6O3, C4H2O4, C5H10, 5C6, H12O6. So 6 hexose is, pentose is 5, tetrose is 4, triose is 3. So yeah, and this is your hydrogen atoms, double the amount of it: 3, 6, 4, 8, 5, 10, 6, 12. And your carbon and oxygen are of equal amount. So you can say that it is a CnH2n ON. This is the general formula. So this is called as glyceraldehyde, erythrose, xylose, ribose, ribinose, glucose, galactose, mannose. So various names of these things. And this is for the aldoses, this is for the ketoses if the ketone group is dominant: ribulose, fructose, erythrose, dehydroxy, acetone, and so on. So let's see these hexoses. That's the main like important one. The third one, C6H12O6. Yeah. Yeah, these fourth. So glucose, the essential energy source of all body functions, and names are dextrose and blood sugar, and components of each disaccharide. So, and the galactose, they are seldomly occurs free in nature and binds uh, with glucose to form sugar in milk or lactose actually, and only absorbed by the body. So galactose is converted to glucose to provide energy. Whereas the fructose is the sweetest of all sugars, 1.5x sweeter than sucrose, and occurs naturally in fruits, honey, than the fruit sugar. So this is the glucose structure. That is your dextrose, blood sugar. Yeah. So here is the first carbon, second, third, fourth, fifth, sixth. Yeah. And this is the another form of galactose. So the major difference between glucose and galactose as the fourth carbon, your hydroxyl group is on the different side. And the fructose is a 5 carbon like pentagon, it's not a hexagon, but it still has 6 carbons. Yeah. And it has these ketose groups, CH2OH, CH2OH, here attached, and it has this O group here. That's the major difference between your three sugars. Now discuss the stereochemistry that we discussed like in amino acids, left hand side and right hand side, like D glyceraldehyde, L glyceraldehyde, D form and L form. So we also call them as optical isomers, that is enantiomers, which differ from each other in disposition of the various atoms or groups of atoms in space around the asymmetric carbon atoms. So they are in fact the mirror image of each other, and they like to be left-handed or right-handed gloves. So one form in which H atom at carbon 2 is projected to the left side and OH group to the right is designated as D form, and where the H atom is projected to the right side and OH onto the left side is called as L form. So this is OH group, and this is also OH group. So this is on the right hand side in the D glyceraldehyde, and in the L glyceraldehyde it's on the left hand side. So for example, glyceraldehyde has been shown in this example in this 2 isomeric form, left and right hand side. So it's like a mirror image. If you put a mirror here, so mainly this one is getting changed. So your D-glyceraldehyde containing D aldoses, is aldotrioses, 3 carbon atom. So we made this D-glyceraldehyde, this 3 carbon atom structure. We can make also 4, 5 and 6 atoms to it. So there will be different structures, that is known as aldotetroses, aldopentoses, aldohexoses. So it will be called as deerythros if it on the right hand side, and D-threose if it is on the left hand side. Then this is D-ribose, D-arabinose, D-xylose, D-lysose. And this is D. You should not go into too much detail, but it's just good to know that how these structures are being divided. D-altrose, D-glucose, D-mannose, D-iodose, galactose and talose, and so on. Just a second, students. Something mischievous is happening. Okay. So now let's discuss some properties of these monosaccharides. That is, they show the property of mutarotation. That is, whenever monosaccharide is dissolved in water, they show an optical rotatory power. So which shows a constant value. For example, if D-glucose is dissolved in water, a specific rotation of plus 112 degree of Celsius is done. But this is quite slow change. So this might be that 24 hours the value will become from plus 112 degree to 52 only. So this gradually change in the structure, half the degree changing of the glucose is known as mutarotation. And this phenomena is also shown by another sugars like pentose, hexose and reducing disaccharides. Second, glucoside formation. So whenever the D-glucose is treated with HNO3